Ichthyosis. Probably you have never heard about it, but for thousands of people it has been a part of their daily life since birth. One of those people is our friend Henke. Listening to his story inspired us to focus our project around this disease. Learning from him, we found out that as of now, the disease has no cure and the daily treatment steals several hours from each day of the patients. We refuse to accept that. And here's where our project comes into play. IchtyoCare is being developed to offer a substitution therapy that could minimize the time patients need to spend taking care of the symptoms. Even though it doesn't offer a cure, it could greatly enhance the quality of life of those affected.
Ichthyosis is an umbrella term for skin diseases that are genetic in most cases. There are multiple of different types differing in cause, severity of symptoms and the age of disease onset. The condition usually results from a mutation in one of the 50 proteins that are involved in the formation of the skin barrier. We have focused our work around 3 genes whose mutation results in the development of autosomal recessive congenital ichthyosis, ARCI. Expressing the genes and producing the active forms of defaulted proteins in vitro would allow the delivery to the skin to make up for the malfunctioning ones. The genes we have chosen are TGM1, ALOXY3 and ALOX12B, respectively encoding the enzymes transglutaminase 1, ELOX3 and 12R-LOX. These specific enzymes were chosen because they are fully or partly cytosolic, which makes them easier for us to begin working with. TGM1 seemed like the hardest one to tackle in the lab, as it is the largest of the 3 and the most complex in structure. But we still wanted to try, as this is the one which causes Henke's phenotype. As our project advanced, we have decided to prioritize ALOX12B. ALOX12B encodes an enzyme which catalyzes the oxidation of arachidonic acid to produce 12R-HPETE. This signaling molecule plays a key role in the lipid barrier formation in the epidermis, essential for maintaining skin hydration. And to transport the active enzymes to the target destination in the cell, we have chosen to develop a delivery system using liposomes as enzyme carriers. Liposomes are spherical, bilayered and hollow molecules composed of lipids ensuring biocompatibility. For our purpose, we have prepared liposomes composed of 50 mol% soy, 25 mol% dope and 25 mol% cholesterol. Dope is a substance easing the liposomal fusion with the cell membranes, while cholesterol stabilizes the structure. Upon reaching the cell, the liposome will fuse with the cell membrane and release its enzyme content into the cell. The project idea, put shortly, is to produce an active form of 12R-LOX encapsulated in a liposome and store them in a serum to be applied onto the skin to allow skin penetration and fusion with the cells. The enzyme then gets released and substitutes the malfunctioning ones.
Our main goal is to produce a cosmetic product called IchtyoCare and raise awareness about the disease among the society. This can be achieved by completing the following goals we have set for ourselves. These include assembly of a plasmid, expression of the protein, purification of the protein, producing liposomes, encapsulation, suitable medium testing, activity testing, and raising awareness and educating about the disease. Additionally, every iGEM team is required to contribute to the future iGEM teams. We have decided to study a chromoprotein called A-blue, hence the following goal: characterization of A-blue.
In human practices, we began our project by reaching out to Ichthyosföreningen, the Swedish Ichthyosis Association, to get input and help on our project. We first got in contact with Agneta Gonemo, who initiated the formation of the association, and she is also an expert in the field. In the same meeting, we also met with Mikael Gonemo, who is president of the association. He is also Agneta's son and suffers from lamellar ichthyosis due to mutation in the TGM1 gene. We've had a long collaboration with them and have learned a lot of useful information in the process. We also received a book from Agneta, written by Agneta, which has been a very useful source. This collaboration motivated us to find a better and more efficient treatment option and to spread awareness about the disease. In addition to Agneta and Mikael, we also reached out to different people to get input on different parts of our project. Mikael and Henrik provided us with the knowledge of how it is to live with the disease. Agneta and Hans filled us in on the evolution of ichthyosis research in Sweden, and Alexandra and Daniel helped us design and understand the liposome as a delivery system. Alexandra also helped us significantly in making our liposomes. We are very proud and grateful for everyone's input in making our project IchtyoCare.
To produce 12R LOX, we began by cloning a LOX12B, the gene encoding the enzyme, into a chosen vector PET28. When the plasmid was complete, it was firstly transformed into one E. coli strain for amplification and then into another for protein expression. The enzyme was harvested using sonication and run through a NI-NTA column and a size exclusion chromatography to purify the enzyme. Further measurements were then conducted to assess the structure and stability of the enzyme. To ensure that we had successfully incorporated the gene into the vector, it was sent for sequencing, which confirmed that the sequence for the plasmid was correct. Afterwards, the expression of the enzyme was induced and tested using SDS-PAGE, which showed 3 bands at a molecular weight close to that of 12R LOX, but there was still contaminants present. We therefore performed a size exclusion chromatography. The SDS-PAGE after still showed the 3 bands, but with all smaller fragments gone. A dot plot also confirmed that we had managed to obtain an enzyme with a his tag. To further confirm whether protein of correct size, a mass spectrometry measurement was performed, which showed a molecular weight close enough to 12R LOX to deduce that we had the correct enzyme. To assess the secondary structure of the enzyme, we used circular dichroism. The structure percentage coincide with the predicted structure obtained from AlphaFold, clearly showing our enzyme had the correct secondary structure. To investigate whether our enzyme had any tertiary structure, we started with a wavelength scan for the tryptophan emission. The spectra showed a clear peak at 340 nanometers, showing that the protein is folded. To further determine the tertiary structure, as well as the thermal stability, a nano-differential scanning fluorimetry was performed. This showed that at pH 7.4 in a PBS buffer, the enzyme had 2 unfolding events, one for each domain. This clearly indicated we had a folded protein. To try to simulate a more native state, we did another nano-differential scanning fluorimetry, but in tris buffer at pH 7. The buffer also contained iron and calcium ions, which according to previous studies, 12R LOX interacts with. The presence of these ions showed an overall increase in stability for 12R LOX.
Construction of the vector and transformation leading to expression of OI blue followed the same lab workflow as a LOX12B, the only difference being that OI blue was purified in one step using a NE-NTA column. Colored appearances of the bacteria culture confirmed the expression of the protein, even before we had induced it. The purity of the molecule was confirmed with an SDS-PAGE showing a single band at the correct molecular weight. The method used to create our liposomes is called thin film hydration and extrusion. For our project, liposomes with a diameter of 200 nanometers were assessed to be suitable, so we used membranes with a pore size of 0.2 micrometers while extruding. To verify the successful creation of our liposomes, dynamic light scattering measurements were performed. During the summer, several batches of liposomes were created and all of them had a diameter of 200 nanometers. Additionally, the shelf life of the liposomes was investigated by measuring the same batch of liposomes across multiple weeks. From the experiments, we saw that the composition you can see in this picture was intact for the longest time. It could be concluded that the liposomes alone had a shelf life of at least 4 weeks, and OI blue encapsulated liposomes had a shelf life of at least 3 weeks. After encapsulation of OI blue and 12R LOX into the liposomes, the protein remaining outside of the sample was removed using size exclusion chromatography, allowing us to have samples with purely OI blue and 12R LOX encapsulated liposomes. The purified samples were analyzed under a microscope in bright field, and the microscopy showed presence of liposomes and OI blue. As the SEC column removed all OI blue outside of the liposomes and OI blue was present in the microscopy, combined with the previous microscopy findings, we found it convincing enough to suggest that the encapsulation of OI blue was successful. Furthermore, as 12R LOX is quite similar in size to the tetrameric form of OI blue, and as we used the same method to encapsulate it, we could deduce that we managed to encapsulate 12R LOX as well. Nano-differential scanning fluorimetry was used to investigate if the encapsulation of liposomes affected the stability of OI blue and 12R LOX. After doing the experiment, it was clear that the encapsulation had no effect on the stability of the proteins as they had similar melting temperatures. Furthermore, aggregation analysis was also conducted on OI blue, and once again, the liposomes did not have any effect on the protein.
Beyond the lab work and the creation of our product, we have also put substantial effort into completing our goals of spreading awareness and educating about the disease. This is something we believe to be extremely important, since our outreach has given us insight into the sad reality of how it is to grow up with the disease, the lack of understanding from those not affected, the lacking aid, and sadly the bullying that follows being different. Now, to our end of helping educate and spread awareness, this work began in the early summer as we participated at the university's Summer Week for Teenagers, where we hosted lab sessions and simultaneously told them about ichthyosis and IchtyoCare to help them understand the severity of the disease. We continued on this track by visiting and holding lectures at the local high school where we taught the teens about synthetic biology and iGEM, where we even got the high school interested in possibly participating in iGEM next year, but most importantly, we talked to the teens about ichthyosis and how people's lives are affected by the disease. Throughout our project, we have also held a podcast to inform about the disease and our progress with IchtyoCare, as well as the progress of other teams. While participating in both the Swedish and Nordic iGEM conferences, we invited teams to the podcast where they informed on their projects and we shared what we have learned so far. Furthermore, at the Swedish iGEM conference, all the Swedish iGEM teams undertook an initiative to teach synthetic biology in a fun way. This resulted in the informative tabletop card game Plasmid Assembler, which you can download on our wiki. We also wanted to leave a little gift for future iGEM teams considering lipid-based nanoparticle delivery systems. Therefore, we, alongside other iGEM teams, together wrote a lipid delivery handbook to simplify the work for future years. This can also be found on our wiki. Safety concerns are always something we considered for our project. We have stayed in ordinance with our university's guideline for a safe lab environment while being mindful to not overuse resources. Furthermore, harmful chemical compounds were taken care of not to be spilled into the environment, but sent to be handled by suitable facilities. For our project, we have looked at the ethical considerations one would have to take, both for what we have done and what we would do for the future. While working with a protein that is to be inserted into a living person, one always has to be mindful of the interactions that may arise from this external insertion. In the case of 12R LOX, its complete interactome is not known, though from a wide array of studies it's been found to be present in several different cell types throughout the body. Therefore, a more thorough deep dive into the specifics of 12R LOX interactions needs studying before any testing can take place. And before any clinical studies, animal testing would of course have to be done firstly, and would most likely be done using mice as has been used in earlier studies for ichthyosis. For this, we would have to follow adequate legislation for animal testing that is present in the country at that time.
This brings us to what remains to be done in the future for our project. If we had more time and resources, we would want to start off with the enzyme activity testing to confirm that the enzyme we have produced is correct and active. This would be followed by more extensive testing of the stability of the liposomes in various media, as well as simulating the liposomes fusion with the mammalian cells. Additionally, we would want to research the unspecific release of 12R LOX from the liposomes and its effects on the non-target tissues. If all the steps listed above turned out to be successful, the project could advance to animal studies and potentially lead to clinical trials. To conclude all that and circle back to the goals we have introduced in the beginning of the presentation, we can clearly see that we have accomplished most of our goals. So, the assembly of the plasmid was confirmed with sequencing, the expression of the protein was confirmed with an SDS jar, the purification of the protein was confirmed with a dot plot, producing of the liposomes was confirmed with measurements such as DLS, fluorescence microscopy and spectroscopy. When it comes to the encapsulation of the protein in the liposomes, we have leads showing that the encapsulation was successful through measurements such as bright field microscopy. Suitable medium testing unfortunately couldn't be accomplished as the medium we have chosen interfered with the DLS measurement. We also couldn't accomplish the activity testing as our resources didn't allow us to use a compatible assay. Spreading awareness and educating about the disease was accomplished through our human practices, education, school visits, podcasts and collaborations. Having said all that, we would love to see IchtyoCare help those suffering from ichthyosis, making their daily treatment at most a weekly treatment.